to our second speaker, Ezgi Karaca from Turkey. So, Ezgi, we were just to give a short introduction. We already actually hosted Ezgi also one of our previous journal clubs last year. So it's really nice to have her again at our Fragile Nucleosome, now in seminars. So actually, Ezgi started in my current university, in Boğaziçi University in Istanbul, in chemical engineering department, working on protein structures. And then upon finishing her master's here, she moved to Netherlands in Utrecht University, where she worked with Alexander Bonvin as part of her PhD studies in computational structural biology. And upon graduating in 2013 from PhD, she moved to EMBL in Heidelberg. And yeah, so since it's actually it's computational biology, I thought it was the other side. So she moved to Heidelberg, where she was working with jointly with Barabas and Carlo Magno groups. And since 2017, she moved back to Turkey and to the beautiful city of Izmir, where she started her own group as computational structural biology. And little by little, now she has moved toward studying the computational structural biology in gene regulation and lots of chromatic modifiers. So, and yeah, floor is yours.
Thank you very much, Ündüz, first to you all for the invitation, as well as for the nice introduction. So indeed, today I will be talking about one of our projects, one of the big projects in my lab, that is on how pioneer transcription factor recognizes its cognate sequence on the compact nucleosome. But before going into this, as Ündüz said, we are located in Izmir, and this is my group. So we are a rather small group, but interested in understanding the structure dynamics and function relationships of biomolecular complexes within different biological contexts. We are working at 3 different levels, and which are sequence, structure, and function. And to link the relationship between these 3 steps or 3 pillars, we use the most suitable methodology, let's say. And within this perspective, we combine integrative modeling, where we guide the complex building in silico along the known experimental data or acquired experimental data. We obviously, of course, use deep learning-based methods, specifically AlphaFold, as I am sure that all of you have been using since the late 2020. And we also heavily use molecular dynamic simulations to be able to understand the conformational changes that happen upon binding, as well as the specific intermolecular interactions. So we trace intermonomer interaction profiles to be able to understand the specific recognition mechanism or selective recognition mechanism between protein-protein and protein-DNA systems. So as I said, we work on different biological systems. So regarding the specific protein recognition or specific protein complex formation mechanisms, within the concept or context of epigenetics, we are specifically interested in understanding the de novo methylation process that is happening in a very highly specific manner, and as well as the action of pioneer transcription factors in gene regulation. And as you all know, pioneer transcription factors have been like a hot topic or a very interested topic in the chromatin biology field already for many years, but only recently we have started to gain atomistic resolution information on these systems, which is very exciting, of course, for all the people who are interested in the structures of these systems. So the idea of pioneer transcription factors is that they can recognize their cognate cognate sequence, their recognition sequence, which is a short stretch of nucleotides on the compact nucleosome, unlike the other transcription factors, which needs an accessible and free DNA. So the idea is, from structural point of view, the idea to understand how PTFs recognize their sequence on the compact DNA is appealing because, as you see, the compact nucleosome is a very dense environment, and it's very interesting. It has been very interesting for me to understand how this DNA binding domains can find and bind to their recognition sequences on in such a crowded environment. So to study this phenomenon, we have focused on a particular protein family, which is a SOX family. SOX is also a very famous transcription factor, as you know. There are 20 SOX proteins identified in vertebrates, more or less 20. So they are of different lengths, and they are really essential to different types of genesis stages, let's say. So they are of different domain content or context, but they all share the common DNA binding domain, which is the boomerang shape SOX DNA binding domain, as you see over here. This DNA binding domain is 70 amino acids long, and it's classified as HMG, high mobility group binding domain. And the specialty of this domain is that, unlike many transcription factors, it binds to the minor group. So instead of binding to major group, it is able to find its recognition sequence on the minor group. And for this, all SOX DNA binding domains, I mean, I will just refer to the DNA binding domain of SOX from this point and on when I'll say SOX. So in all SOX proteins, you see these critical amino acids, which are strictly conserved in all the SOX DNA binding domains. And thanks to the arrangements of these amino acids, and thanks how they recognize this specific 4-nucleotide TTGT motif, SOX is able to induce a large rearrangement on the DNA and specifically recognized through hydrogen bonds by these polar amino acids with the given TTGT motif. So if we dive into further how SOX can bind to a minor group, which is a very shallow surface to bind, we'll specifically see that besides these polar amino acids, these two hydrophobic amino acids, which are phenylalanine and methionine, are actually embedding themselves into the recognition sequence so that the charged DNA backbone is being sort of distanced from each other. And this creates a landscape or an environment or a compatible electrostatic environment for the protein to bind. So in 2020, the first SOX-bound nucleosome structure was released. It was resolved by electron microscopy, and already at that time we started to work on this project, and that's why it was very exciting for us to see how SOX was characterized to bind to this very narrow minor group. And in this study, given the double-stranded nature of the DNA, the TTGT motif was located or placed on the histone-facing site of the double-stranded DNA, on the specific superhelical location, which is called as SHL2 on the nucleosomal coordinate system. So after seeing this structure, which indicates that the tip of the boomerang is basically clashing on the other parts of the double-stranded DNA on the nucleosome, and because of this, or thanks to this, it induces a structural opening of the double-stranded DNA. So after seeing this binding mode, we asked ourselves, okay, if this is valid, and obviously it is valid, and we see how SOX opens up its sequence on the nucleosome and binds to it and induces a specific DNA opening on the other double-stranded DNA. We asked ourselves, what if we place the SOX binding motif on the opposing strand? Because in principle, given the robustness of the mechanism induced by SOX, the SOX binding motif should be able to be present on both strands. And our study is specifically focused on exploring how solvent-facing, how the SOX binding will occur if its recognition motif will be placed on the solvent-facing nucleosomal DNA. So obviously, this is a very complex system to study, even with experimental methods, so complex that it took many years until the first structure of SOX-bound nucleosome is released. And if something is very difficult to be explored by experiments, it's always also very difficult to be explored by computational methods as well. But still, we were up to this, up to understanding how first SOX can bind and open its recognition sequence, and second, whether with the methodology that we will be proposing, we will be able to elucidate the other type of binding of SOX on the nucleosome. And for this, we combined several cycles of molecular dynamics with integrative modeling. And this is needed because the compact version of nucleosome basically doesn't confer a DNA conformation that would allocate SOX binding. And our idea was to generate a pipeline where we would end up with SOX-bound nucleosome, which will be later tested with experiments by DNA footprinting experiments. So before going into our numeric results, I will just show you a movie which shows the outcome of these cycles of simulations and as well as structural modeling steps. So at first, you'll see how SOX won't fit on its reference state, let's say on the nucleosome, because minor groove is minor groove and it's not wide open enough so that SOX can be accommodated. And after our simulations, we are able to capture some conformations that are wide enough to have a sort of initial encounter complex formed by placing SOX at its place. And then after some other simulations, we observed that the real opening is actually induced by SOX binding with these with the placement of these two hydrophobic residues. Again, at the SHL2 position, spherical location 2 position, as it was resolved, because we know that SOX is going to bind there, as it was resolved by the experiments. So what we did was, in the end, after establishing our model, we checked two things. First, whether throughout our molecular dynamics simulations, we will be able to capture SOX DNA shape in different conditions, and as well as SOX binding fingerprint profile. So here on this slide, you see our shape metric, our shape check, which we defined as phosphorus RMSD. So we basically measure the phosphorus RMSD of the DNA to the initial SOX-bound DNA conformation. Here we see the distribution of all the DNA conformations throughout our modeled bound simulations. And as you see, they fluctuate, so DNA conformation fluctuates when it is bound to SOX, the free DNA, around 1.2 angstrom or so. And when we simulate the free nucleosome, where we incorporate the SOX binding sequence at the SHL2 positions, as you see, we see only, as I said, a couple of conformations that are exerting an RMSD less than 2 angstroms. And when we model with integrative modeling, when we place the SOX at its binding spot on these conformers, and then perform another set of simulation, we'll see that we achieve the RMSD distribution as in the free DNA. So this was very surprising positively surprising for us, because with this we understood that also when we place the binding sequence of SOX on the other strand, we are able to reproduce the SOX shape as in the free DNA bound form. And besides this, we also trace the specific or non-specific binding profile of these special amino acids with the target DNA sequence, because we know that the interaction profiles is in the free DNA, as you see in the blue ones, should be reproduced also within the nucleosomal context if we are going to assume that we are going to have the same recognition pattern. And indeed, this is the simulation, this is the recognition, like a fingerprint interaction profile on the nucleosome at SHL2. And as you see, along the timeline from 200 nanoseconds to 500 nanoseconds, we are able to reproduce the same binding profile. So this tells us that actually it is also possible to have the solvent-facing SOX binding. And in this setting, we are able to fit perfectly well SOX, which tells us that in this binding mode, we wouldn't expect any large structural rearrangements. And this is something this is something, of course, experimentally testable. And on top of this, it also tells us that there should be more beyond the structure that was already published. Sorry for the worsening of the resolution over here, so the image got sort of corrupt. But as the second step, after being sure that SHL2 is a binding location for SOX, and then it can accommodate the other type of solvent-facing type of binding as well. After this, we asked us whether we could perform the same pipeline, same computational pipeline, if we assume that SOX is going to bind at SHL2, SHL0 at the diet position or at SHL4. So we basically went 2 SHL positions to the left and 2 SHL positions to the right, so that we make sure that we are far away from SHL2, but still we are going sort of incremental to have a decent test of our protocol. So at the diet position is over here, SHL4 position is over here, and SHL2 position was over here. And when we perform this, perform the same set of modeling scenario and also simulated the finally acquired complexes, where again, the SOX binding sequence is on the solvent-facing strand. But we observed is that assuming that the SHL2 nucleosome binding of SOX is our reference, because here we see, as I said, we see the same level of fluctuation of DNA shape as we observed in the free DNA bound SOX. And as we see, as we compare the other positions, we see that the SHL4 position is it can accommodate SOX binding, but it seems that it's not going to be as effective as at the SHL2 position. And if we check the diet, it doesn't seem that we will have a specific recognition over here, assuming that shape is one of the factors of specificity in the system. And if we compare again the fingerprint interaction profiles, here we show the specific amino acid nucleotide interaction profiles, though the nucleotides are not specifically noted over here, but we only mark the specific interactions here. As you see, interestingly, the hydrophobic interactions are sort of conserved, but it looks like methionine is making more frequent interactions compared to the reference profile. And we also see that these specific polar hydrogen bond forming interactions, they are lost, and they are totally lost, for example, for 2 at SHL4. And for this, for the diet, tyrosine 118 is happening in on and off manner. So to be able to experimentally test our hypotheses, that we drew from these results, which is that SHL2 should be the most optimal binding site for SOX, and then the second optimal binding site from the shape analysis would be SHL4, and diet should be the least probable binding site. To test this, we worked in collaboration with Dimitrov Lab, and Dimitrov Lab is an expert in using UV laser footprinting experiments. So they are basically imposing UV lesions on the free DNA and protein-bound DNA, and then they compare the footprinting profile of these two states to be able to understand at the single nucleotide resolution where the protein is binding, because the altered nucleotides, and this can only be measured at the level of G, because the DNA lesion is being imposed at G's. So they can observe which G's are being protected, or which G's the chemical environment is altered, etc. And based on this, they can say, okay, these G's are specifically taking part in binding, or the environment is altered, which is going to tell us whether there's going to be a specific binding given the protein of interest or not. So we have different experiments, of course, but I'm just, for the sake of time, I'm only gonna present the final experiment, which is supporting what we have observed during our computational protocol. And this is the UV laser experiment, where the SOX binding sequence is inserted at the same place where we inserted them on the modeled nucleosomes, and three of these positions in this experiment are exerted on the same nucleosomal DNA. And as you see here, we have the diet binding site, here we have the SHL2 binding site, and here SHL4 binding site. And if we quantify these gels, we'll see that at SHL0, meaning the diet, we don't see any specific binding at all. So there is no shape change or chemical environment change for this position, which is in line with our RMSD observation. And we also see that these are apparent KDs, obviously, so they are not real KDs, but we still, with our apparent KD calculation, we see that SHL2 looks like to be a more appealing binding site for SOX than SHL4. And this is also quite obvious with the disappearance of the band widths. So in the end, so we have more analysis on this, quantitative analysis as well, but I didn't put them over here for the sake of time. In the end, our hypothesis is that at SHL2, SOX can bind as if it binds to free DNA, because the histone environment over there is allowing the most space so that its electrostatic environment, and also its special environment is least affected. And as you see, so we marked here the let's say non-occupied area by both the DNA, histones, and SOX. And as you see, the largest space is found at the SHL2 position. This also explains why in many high throughput experiments, people found to have SOX bound specifically at the SHL2 sites instead of at other positions. And that's why I assume, or I suspect, that the available structures of SOX-bound nucleosomes so far, they contain either the SOX bound at the SHL2 or minus SHL2 position. And there's another structure, but in that scenario, SOX is bound towards the end of the nucleosomal DNA, and there is more free to detach or to be recognized. That's why this may not be considered within this context. So if you are curious about this work, you could go and check our preprints. This work is right now under review, and I hope that soon it's going to be out with some additional analysis. And of course, I would like to thank here specifically Burcu, with whom we have been carrying out most of the computational work, Berçin as well. She is also from my lab, and she has been helping us out in several simulation setups and analysis. And of course, Stefan Dimitrov, because he has been the one who has introduced us into his experimental collaborators, so that we will be able to dive into the impressive world of nucleosomal recognition. And if you are interested in this work or in other related work, you can follow me on Twitter, or you can always send me an email. Also, if you would be willing to join our lab, you can send me an email. We are always interested to have motivated students in our group. Thank you very much for your attention.
Thanks, Ezgi, for this great talk. If you have any question, you can raise your hand, or you can write in Q&A box, so we can go for it. I think Ben wanted to ask a question. If you want to go, Ben.
Thanks. That was a great talk. I really enjoyed that. It's fun with the Differential nucleosome because we had Alicia and Michael presented on the structures of SOX and OCT4 binding to the nucleosome a couple of years ago. And so this kind of revisit the same topic from another approach is really fantastic. I was wondering, how do you think, like, your simulations and the results for where SOX is binding to the nucleosome, how do you think that would change with if the nucleosome has like a histone variant incorporated or histone post-translational modifications?
So I have to say here that in our simulations, we didn't take into account the histone tails. The major reason for this is that we want to make the system as simple as possible, so that we will be able to concentrate only on the DNA recognition by SOX and like the localized histone environment. But obviously, expanding on what we have found, one could test the same setting by using histone variants or histone tail modifications, because surely they also encode biophysical signals, and those signals should have an effect in the dynamics of the nucleosomes. But whether that's going to be independent of the SOX DNA binding or not, that is open for further exploration.
And I guess the other question I have, and I ask this for everyone who does nucleosome work with the 601, is how do you see these kind of like requirements for the different helices for where it's binding? How do those change if instead of being like a really well-positioned nucleosome, it's more of a natural DNA sequence?
So to this, we also asked ourselves the same question. So the main reason why we used 601 was because we planned that what we will be finding will be tested by experiments. That's why we didn't want to have any sequence effects that's not going to match what's going to be observed in the experiments. But to answer this question, we also simulated a natural DNA sequence by having the SOX sequence inserted at the SHL2 position. And interestingly, the RMSD profile or general nucleosome dynamic profile was the same as in the 601 case. So in the case of SOX, it really seems that somehow, as long as SOX is close to its binding site, it's going to open it itself, irrespective of the general DNA sequence. So that's at least our observation.
Awesome. Thank you.
Thanks.
Thank you, Ben. Any other question? I think we are also running out of time. So, okay, then I think that I will wrap it up. So thanks for the great talk by Adi and Ezgi. And thank you everybody for joining. Hopefully we will see you in 2 weeks time in our next session. And thank you.